Hey dear learners, I welcome you all again to your very own YouTube channel LavEd. I am your host Lavnish and I am very excited to help you understand your class 12 biology and very excited to continue our journey through NCERT biology line by line with you all. In the last video, we explored the introduction and the pre-fertilization structures and events of the chapter Sexual Reproduction of Flowering Plants. And today we are going to dive into the next topic, that is the male reproductive system or the male reproductive parts in flowering plants. Okay, but before we get started, make sure that yesterday the points I told you, you should be prepared with that. Number one, sit with your NCERT book open, pause the video after I teach you a para, go through it by yourself, make the concepts clear, and then resume the video and repeat it. Okay. Number two, biology is a very volatile subject. Revise the content very regularly. And number three, practicing UIQs on the topic that I teach you. If you follow this, I guarantee you will rock. Okay. So also at the same time, I want to make you clear that do completely follow the NCERT NEXUS series, because we will be covering NCERT line by line, each and every line in very detailed explanation according to your latest rationalized syllabus. Without any further delay, let's dive in to our today's session. Be ready with your books open. So today, again, I have highlighted today's NCERT also. You can highlight it in your NCERT, or either you can take notes and write topic wise in your notebook, because at the time of the exam it is the only thing that you should do, to go by the points that you have already made during the lectures. It is not before the exam, it is not a time where you actually read the NCERT. It should be like that, only we're highlighting the important points and recalling them before the exam. Okay, so these are certain tips as a senior I would like to mention you all. Okay, so let's dive into this session. So today's session, the topic is mainly on, you can say, the male reproductive system of flowering plants, known as the androecium. Okay, the now we should know what is androecium. Okay, I have written over here line. Androecium consists of whole of stamens. Okay, wait a second. I'll show you what is a stamen. Okay, this structure you can see right here. Yes, this combination of anther and filament is called as stamen. Okay, so what is written? Figure 1.2a. This figure shows the two part of a typical stamen. Okay, I have already mentioned stamen is made up of two part: the elongated tube-like structure, the long and slender stalk called the filament, and the terminal generally bilobed. Terminal मतलब क्या होता है? Terminal means the part which is far away. Okay, proximal means part which is close. Okay, so the terminal generally bilobed called the anther. The terminal one is called as anther. Okay, and the long slender stalk is called as the filament. Okay, now what is given? Next line. The proximal end of the filament is attached to the thalamus or the petal of the flower. Okay, look, I can show you yesterday's diagram only. Okay, you'll be very clear at that. Proximal. What is proximal? Proximal means near. Okay, the near end. This is the near end. Okay, this is the near end. Look at the cursor. The proximal end of the filament is attached to the petal of the flower. Petal. This is the petal. Yes, this part. The proximal, because we always measure the distance from foot to height, not from top to bottom, but from bottom to top. So the bottom is close, so hence it is called as proximal end of the filament. Clear? Okay, next point. Now this is an important line because it is mainly asked in MCQs type. Okay. The number and length of stamen are variable in flowers of different species. Okay, it is a mainly, what can I say, it is a fact that you should remember, especially when questions come or like statement type question comes, statement 1, statement 2. Okay, then you should remember, yeah, the number and length of stamen variable. Clear? And this is nothing but what? Useless thing. Okay. If you are if you were to collect a stamen from each 10 flowers, you can read by yourself, nothing that important. The important points already highlighted. Okay, you should be very, 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 very focused on that points. Clear? Okay. What is good? Now this para is mainly regarding the characteristics of anther. Okay. Filament, no such characteristic. Only thing you should know, there is a long slender stalk. Now anther being the hub of the androecium, or you can say stamen, more points are to be discussed on the topic. Clear? Look, a typical angiosperm anther is bilobe. Okay, what is bilobe? Number one, what is bilobe? Bilobe means two lobe. You can see this part. This is one lobe. This is the other lobe. So hence it is called as bilobe. Okay, with each lobe having two theca. Now what is theca? Theca is nothing but the two cup-like structures. You can see this. If I consider, wait a second, I will draw. Okay, for you. Okay, my drawing will be bad. This part. Okay, this separate the two part. Okay, this is one loop, this is the other loop. Okay, now you can see out here two cup-like structures, two lobes. This is one, this is the other. Okay, these two. Now it's important that because each lobe consists of two cup-like structures, hence characteristic क्या होगा? That they are diathecous too. Okay, now wait, I'll change the color. What is given? Often a longitudinal groove runs lengthwise separating the two theca. Means what? You see this part. Longitudinal groove running lengthwise. Okay, it is cutting like that. Longitudinal it is cutting the two thicas, or you can say a cut in a single lobe in longitudinal manner, which separate the two thicas. Okay. Next page there is a diagram. I will show you. Okay, but let's focus on what is given. Let us understand the various types of tissues and their organization in the transverse section of an anther. Okay, the bilobe nature of an anther is very distinct in the transverse section of the anther. Anther is a four-sided tetragonal structure consisting of four microsporingia. Okay. Now remember the shapes. Okay. Anther, tetragonal structure. Later we will discuss the structure of microsporingia. Then we will discuss the structure of shape of pollen grains. So keep in mind, anther is tetragonal. Okay. Anther mainly. Okay. This lobe, whole lobe consists of four microsporingia. You can see one, two, three, four. So these four microsporingia, which are located at the corners. Okay, they are not located in the centers. In the next para, we are gonna study the structure of microsporingium. Okay, how they are located, what are the layers. Okay, we are gonna discuss. But first of all, let's make it very concise. Anther, yes, anther, tetragonal structure. Clear? It's like a tetragon. Yes, looking. Okay. Now it consists of four microsporingia. One, two, three, four. Two in each lobe. Clear? That is the main thing. One more time. Anther, tetragonal structure. It is a bilobe structure. Each lobe consists of two cup-like structures, hence called as didicus. Now, two cup-like structures, also called as, not also called, but cup-like structures, each cup-like structures consist of microsporingia. Hence, in a lobe, there becomes two microsporingia. Okay, that is the main thing. Let's head to the next point, next para. Okay, wait a second. Okay. The microsporingia develop further and become pollen sacs. Now look, this circular structure, okay, is basically microsporingia. They later develop to pollen sacs. Now the pollen sacs mainly consist of pollen grains. Is it clear? Anther, two lobes, inner lobe, two cup-like structure. A cup-like structure consists of microsporingia, microsporingia developing to pollen sacs packed with pollen grains. Repeat by yourself one time. Okay? Okay. Now we are gonna discuss the structure of microsporingium. Okay. I think I can make it clear to the next page also. We'll see. Yes, this is the main thing that I was talking about. Okay. I'll come to this diagram, but I think, okay. First, I think I will discuss the diagram only because then theory will be easy for you to understand. Okay. Look, first of all, the microsporingium are circular. You can see the circle. So they are circular in nature. Clear? These usually consist of four walls. Look, if we zoom this, we'll get first layer is the epidermis, then we'll got endothelium, then we get middle layers, then the last layer becomes the tapetum. These are the four layers. Okay. Now this three layers main function is provide protection and prevent from dehiscence of the anther. Okay. Dehiscence of anther, not prevent dehiscence, but dehiscence of anther. Okay, promote. Now, tapetum function is very important. It provides or nourishes the developing pollen grains. Okay, being the innermost layer. And this tapetum is very rich in cytoplasm and generally contains more than one nucleus. Okay, now I think we can go to the theory part. The thing that I've told you now will be implemented in the theory part. Very important. Okay, check. So, in a transverse section, a typical microsporingium appears near circular in outline. Yeah. First of all, I told you that anther was tetragonal in nature. Now this time microsporingia. Microsporingia is what? Circular. Clear? Microsporingia. This four dots. Okay, one more time I will show you. This four. One, two, three, four. These four are what? Circular. But the overall structure is looking like what? Tetragonal or not? Four-sided structure or not? So that is the main point they are trying to highlight on this part. Okay. It is generally surrounded by four layers. I have already discussed epidermis, endothelium, middle layers, and tapetum. Okay. The outer three layers perform the function of protection and help in dehiscence of anther to release the pollen. I have already discussed. The innermost wall layer is tapetum. Okay, the innermost layer. Yeah, it nourishes the developing pollen grains. Cells of tapetum possess dense cytoplasm and generally have more than one nucleus. Yeah, I think I have already discussed. Read the para, make yourself very familiar with the diagram, then come again and study. Okay. Now you will get 3 points regarding anther. Number one, when anther is young. Number two, when anther develops. And the last one will be when anther matures. Okay, these 3 points very clear. Make it very distinct. Okay, because questions can be asked again. When anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. Okay, what I'm telling, microsporangium other name. Okay, microsporangium other name. Don't be confused with sporogenous tissue and microsporangium because the other name of microsporangium is sporogenous tissue only. Okay, they are just trying to confuse by giving other names. Okay, that is the main thing given out. Nothing more. You should know. You should know the main difference that sporogenous tissue and microsporangium. That the thing is that there is no actual difference only. Both the terms are same only. Okay. Then we'll go to this point, microsporogenesis. Okay. Now comes, look, I told you anther young. Now happening what? Anther developing. Okay. As the anther develops, the cells of the sporogenous tissue undergo meiotic divisions to form microspore tetrads. Now imagine the situation. Okay. I'll take you back to the diagram. This one. This is called what? This is called what? This goal. This round structure. is called as microsporangium. Okay, microsporangium, other name is what? Sporogenous tissue. Now, each microsporangium or sporogenous tissue consists of tiny, tiny cells. Okay, tiny cells. Okay, like this, like the dots. There exist a lot of cells. Okay. Now, what happened? Each cell, each of the dots are capable of generating pollen grains. Okay. So, hence, each of them, each of the cell is also known as pollen mother cells. Clear? How they do? First of all, let's consider this one dot. Okay, this dot. Okay, this dot. This dot will undergo meiosis one. It will create two dots. Okay, then this will go. These two dots will undergo meiosis two, resulting in each one two, and this one two. Clear? A total of, this is the final progeny that a single pollen mother cells can create. So hence also called as microspore tetrads. Is the concept clear? I know that the drawing is bad because my handwriting is bad, not that good. Okay, but I have also certain limitations. Okay, uh, in online mode. So yeah, that is the only thing. Okay, clear? Should I teach you one more time? Okay, I will repeat it. Look, you got. Just listen to me. Okay, it will be very clear. First of all, you will get the anther. This is the anther, tetragonal in nature. Anther possessing two lobes, hence called as bilobe. Each lobe consists of two theca, right hicus. Each theca consists of microsporangium or sporogenous tissue. Okay. Now, each sporogenous tissue consists of millions of cells. Those cells also called as pollen mother cells because they can give rise to a single pollen grain. Yes. Now, the cell from the sporogenous tissue come out. They perform meiosis one. They generate two cells. They The two form cells again perform individually meiosis 2, resulting again in overall a microspore tetrad. Okay, so that is the main concept out here. Nothing to worry. There is the process of microsporogenesis. Okay, say by yourself. And if you have any doubt, then definitely you can ask in the comment section. Okay, without any hesitation. Okay, let's go ahead. Now what is given? Same thing already written over here. As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential. I have already discussed. Okay, the process of formation of microspore from a pollen mother cell is called as microsporogenesis. If it is bored, you have to write this thing. Okay, you can also imagine the concept. Then also you can write. Okay, nothing to worry about. The microspore, as they are formed, are arranged in a cluster of cells called a, yeah, I have already told the resulting four cells. Okay, also known as called microspore tetrad. Okay, now earlier, I told you anther young, anther develops, now anther matures. So as the anther matures and dehydrate, the microspore dissociate from each other, developing into a pollen grain. Simple. Yeah. We have already discussed the pollen sex develop, matlab, they will give pollen grains. Now similarly, same thing, anther matures. Yeah, it is trying to convey that pollen sex develop and resulting in pollen grains. Same thing. Okay, just trying. The NCERT thing is that it tries to confuse as many ways as possible, but make it very simple. Try to decode the every line. It will be very, very easy. I can guarantee you that. Okay. Now let's go ahead. Okay. Pollen grain. Next topic is pollen grain. The pollen grains represent the male gametophyte. Okay. If you touch the open anthers of hibiscus or any other flower, you'd find deposition of yellowish powdery pollen grains on your fingers. Yeah, you can do it practically. I think you will be already doing in class 12 practicals. Okay. If you are a dropper, then definitely you have, I think, done in your class 12. Or if you are in class 12 now, you will do this time, I think, practical. That you will be given a flower. You will be asked to detach the pollen grains like that. Okay. It looks basically a powder in nature. Okay. Yeah. Nothing else. Just 30. Okay. Look, this para I have highlighted. I have used many colors in order to separate the lines. Okay. Pollen grains spherical. I told you earlier that number one, anther tetragonal, microsporangium circular. Now pollen grain, which is the result of microsporangium after meiosis, is what? Spherical. Clear? The three shapes are very clear. Okay. Pollen grains spherical, measuring about 25 diameters. Okay. It has a prominent two-layer wall. Now I think this is given. Okay. Look, two-layer wall. The outer layer is called as the exine. If I will be able to draw, I will draw. Okay. Just look at the diagram. Sorry, but I think it be done. Drawing the computer is very difficult. Wait, get the screen. So the blue color, the outer line that I have drawn. Okay, wait, I'll erase this one. Let it remain only. Wait. So pollen grains, okay, two layer you look, two one layer. The blue layer you see, that is basically called was exine. Okay, the hard outer layer called the exine, made up of sporopollenin. Now this blue layer is made up of sporopollenin, which is one of the most resistant organic material to be ever known. Okay, it can withstand high temperatures and strong acids and alkali. No enzyme that degrades sporopollenin. Okay, so it means there is a very exine is made of sporopollenin. Sporopollenin is very resistant to any kind of alkalis, acids, or even enzymes. Cannot even degrade it. Okay. So again, you will get, I think, a question of assertion region. You can get very famous question. Look, pollen pollen grains are the question will like that. This will be given as assertion. Assertion: pollen grains are well preserved as fossils. Reason: because of presence of sporopollen. Okay, if you are giving boards, then definitely one more question. Give reason why pollen grains can be stored as fossils. And if in competitive exams you will be given assertion region, thing only remember because. Okay. Now you can see that the diagram I have drawn, I did not draw a circular outline. I have drawn certain kind of arrays. Okay. So in order to prove that the exine, the exine exhibits a fascinating array of patterns and designs. Okay. Why rating is nothing important. Okay, now we'll move on to the inner wall. Okay, look, I'll teach you. Okay, the inner wall. You can see the inner wall. Yes, the black part inside the blue. Okay, the inner wall is made up of what? Cellulose and tin. Okay, it is not like exine where it is disconnected at the bottom. Okay, now disconnected part. This disconnected part is also called what? This part. This part. Okay, this part I'm talking about. This is called as Jaunpur. If I'm not wrong, it is Jaunpur. Okay. Now look, this is inner layer. You can see this inner layer. Okay, this is made up of cellulose and pectin. Okay, make it very clear. This is made up of cellulose and pectin. There is a continuous layer. Nothing much to say. Let's read what is given in NCERT. Okay. So the inner layer of the pollen grain is called intine. Ha. The outer layer exine. The inner layer is intine. Thin continuous layer made up of cellulose and pectin. The cytoplasm of the pollen grain is surrounded. Okay. When the pollen grain matures, it contains two cells. Okay. Now the cells come. Look. This is the vegetative cell. This part. And this is the generative cell. Clear? The vegetative cell is generally bigger in size because its primary function is to provide nutrition. Okay. So hence it has irregularly large shaped nucleus. Okay. And it is mainly used for reserve food material. Okay, that is the only given. I think the vegetative cell is bigger, has abundant food reserve, and large irregular shaped nucleus. Now, if we look at the generative cell, the small cell is basically what? Generative cell. Okay, generative cell is generally smaller as compared to the vegetative cell, and it basically flows in the cytoplasm. Okay, that is the only thing I can say, or given in NCERT, and nothing to talk much more beyond NCERT, because NCERT is the key, okay, of scoring marks in biology. Okay, now this stage is basically observed. This stage, okay, it is observed in 60% of angiosperm when the pollen grains are shed off. 60. Yeah. My handwriting, no, not my handwriting actually. The limitations that I have here, I think you should adjust. Okay, you can read the lines of NCERT only. Okay, look. What? In over 60% of angiosperms, pollen grains are shed at the two cell stage. This is called as two cell stage. Okay, this stage is called as two cell stage. Now, in the remaining species, not in the remaining species, the 40% of the angiosperm, okay, 60, 100, 40. So, 40% of the remaining species, the generative cell, this generative cell is divided into two male gametes. Okay, and hence, since three cells we get, hence it is also called as a three cell stage. Since earlier in 60%, there was only two cell, vegetative and generative, two cell, and three cell because the generative cell divides into two male gametes. Clear? Okay. Next, what is the point? Pollen grains cause. Okay, this is basically what the effect of pollen grain on humankind. Okay. Earlier we talked about the structure of the pollen grain. Now the topic heading will be the impact of pollen grain on humankind. Okay. So pollen grain cause allergies and bronchial afflictions. Okay, that lead to chronic respiratory disorders like asthma, bronchitis, etc. Yeah, it may be mentioned that. Okay, this is an important point, and this is a line that comes in other chapter as well. Okay, it may be mentioned that Parthenium or carrot grass that came into India as a contaminant with imported wheat. So Parthenium or carrot grass, same thing. They came to India as a contaminant with imported wheat. Okay, so that is the only point I think. This is the first impact. Okay, this is the first impact, that allergies, infections, etc. Now let's go to the other importance. Pollen grains are rich in nutrients, hence they are used as food supplements, and pollen consumption has been claimed to increase the performance of athletes and racehorses. Look, this impact, either it can come, you have to read only. Okay, nothing much to explain because these are general lines. Yeah, what I will explain out here. Yeah, basic English, basic, nothing more. Only you can highlight in your NCERT, and definitely you can revise these things. Okay. Once they shed, pollen grains land on the stigma before they lose viability. Now, what is viability? The period up to which the pollen grains can produce the other parts. Okay. So this is already given. The period for which pollen grains remain viable, variable. Okay. Now, when I used to study these parts, okay, I was generally very confused with how many time which one. Okay. So 30 minutes in case of rice and wheat. Now, since the name are smaller, rice, wheat, okay, so you can recall it like that. Since they are small, so hence their viability also small, 30 minutes. But in other members, where names are very big, like Rugiaceae, Leguminaceae, Solanaceae, they remain viable for months. Okay, this is the third application, pollen viability. Then we can also store pollen grains in liquid nitrogen at temperature of minus 196 degrees Celsius. So storing pollen grains have been used in crop breeding programs too. Okay. So four impacts of pollen grains we have studied. Number one, pollen allergy. Number two, pollen tablets. Number three, there is pollen viability. Okay. And number four, storing pollen grains in crop improvement programs. So that was all about today's lecture. Okay. This is the today's topic. I hope you have highlighted the things in NCERT. Okay. I will sum up what we have studied in today's lecture. Okay. First of all, we talked about the male reproductive system in flowering plants. There is the androecium in very detail. Okay. We talked about stamen parts, filament, not so long. Then we move on to anther. Anther, we discussed all the characteristics. Okay, of anther we very well discussed. With all characteristics. Okay, then anthers inside structure. Okay, you can see the microsporangium. Then the microsporangium structure. Okay, then a microsporangium structure. Then the process that happens in them. Okay, how pollen grains develop. Okay, the then pollen grain individual is a topic. Then pollen grain structure. Okay, then pollen grain impact. So this all the subtopics make up the male reproductive system in flowering plants. Is it clear? So you guys made it to the end, and I feel we'll make this series very successful. Till then, keep practicing and stay curious. Thanks for watching. If you have any question, leave them in the comment section below and do hit the subscribe and like button. Thank you.